Yes.
Yeah.
So the next speaker is Jessica Lee from the Department of Statistics, and you're going to have two things you're going to talk about?
Yeah.
Great.
Yeah. Okay. Thank you. Thanks for giving me this opportunity to introduce our software here. The first software I will introduce is called NMFP. So by its name, it's a pre-selection method based on non-negative matrix factorization. The goal is to reduce the search space for possible mRNA isoforms if you want to discover novel ones from RNA-seq data. So first of all, so this is a very brief introduction for ternary splicing. And I think for the audience here, I can skip this very quickly. So assuming that we know the exons of a gene well, then the mRNAs are just combinatorial patterns composed of multiple subsets of exons. So theoretically, if a gene has 4 exons, then there's a 2 to the 4th minus 1, a total of 15 possible isoforms. So you see that as the number of exons goes up, then the number of possible isoforms grows exponentially. So that number can be very large. And alternative splicing is very important because it contributes to the diversity of mRNA isoforms and hence proteins. And also aberrant structures or abundance of mRNA isoforms can cause various human diseases. So the goal for isoform discovery is that, so assuming this is the underlying true isoform, and these are the RNA-seq paired and RNA-seq reads generated from this isoform, the goal is to discover this isoform from your RNA-seq reads, because isoform information is not known, assuming for new isoform, although we know some of them in annotations, but for the new ones, we do not know their structure. We only observe the reads. So the question is, how can we discover the isoforms? So in this setting, we actually calculate the number, a space of possible isoforms by enumerating the possible combinations of exonic regions. So here we define every region between 2 alternative splicing sites as a subexon. So here, these are the isoforms we know from the annotation. And on each isoform, we know the exons in them and their boundaries. But you can see that for some regions, they are only existing in part of some isoforms, but not others. So therefore, we're defining subexons so that by enumerating possible combinations of subexons, we can recapture these annotated isoforms and potentially new ones. This is the parameter we want to estimate. This is the parameter. And for the data, we're categorizing paired and RNA-seq data into bins. So here the bin is defined as 4-dimensional vector of exon indices. So here 1111 means that exon 11, subexon 1111 of the position of these 4 positions. So here there's a paired and read, and we denote the starting position of the 5' end as S1, ending position of 5' end as E1, and for the 3' end, it's S2 and E2. And then we find which subexons these positions are in, and this becomes the bin. So therefore, you can see that as long as we have the subexon number n, we can calculate how many possible bins are here. And then for each bin, we just count how many reads are in each bin. And this is what we call bin count vector. So this is our data. And the formulation of the NMF is like this. So here we have the bins as the rows, and the samples are as multiple RNA-seq samples. The idea is that if we do this factorization, so in this matrix, the rows are still bins, but the columns are reduced to the number of potential isoforms. And over here, potential isoforms as the rows, and the samples as the columns. So therefore, the very good interpretation of NMF here is that, so this W matrix is the composition of potential isoforms, because every column we will require to be binary. So it will mean, okay, whether this isoform contains this bin or not. And over here, it becomes the per column is per sample, the abundance of each isoforms. So this is our ideal thing. So we want to achieve this. But of course, there are a lot of technicality here, because for NMF, first of all, the solution may not be unique. And also, the default version of an NMF does not require this binary. binarized requirements, so we also need to handle this. And also another thing is that although we can reduce the number of columns from number of samples to isoforms, and isoforms are combinations of beans, linear combinations of beans. But the problem is that these may not be true isoform or valid isoforms. So we also need to take into account when a column here has beans that conflict with each other, how do we deal with it? So these are all buried in the framework here. So this is the flow chart of our software. So for this talk, I think I'll skip those technicalities, but just talk about the application results of this method. So first of all, we go through a data normalization step, and to handle the issue that NMF does not give unique solution. So we actually take multiple runs of NMF on the same data. So default is that we run this for 100 times, and in every run, we will process the result, for example, by removing conflicting beans, and also like splitting some isoforms which are not valid into more isoforms, and then we get a candidate pool. And then for the candidate pool from each run, we aggregate them by only keeping the isoform candidates that are reported by at least half of those 100 runs. So that's 50 runs. So for the isoform candidates appearing in more than 50 runs, we keep them as our final isoform candidates. So therefore, we can find the isoform candidates that are frequently selected by NMF over multiple runs. And so we first try the method in some simulation. So in the simulation, the good thing is that we know which isoforms are true. So we set the isoforms before we generate the data. And we use Flux Simulator to simulate RNA sequencing data. And this is in Fly. And so the result I will show here is this. So we actually calculate the precision and recall, and then their geometric mean F-score at the transcript level. So that means we want to see how accurately the method can help with downstream method in detecting the correct isoforms. We also have the result on nucleotide level and exon level, but I think these are not as meaningful as this one, so I'm only presenting this one. So you can see that the nice thing is, first we run just the native cufflinks, so use cufflinks to discover isoforms from RNA-seq data, and then we add NMFP, our method, as a pre-selection step before cufflinks. So you see that here, this is the NMFP plus cufflinks, and I think cufflinks is right here. So the precision is a bit lower. The problem is that we reduce the number of possible isoforms, but you see the recall is much higher. So here, this is the recall, this is that. So essentially, using NMFP can actually increase the chance for cufflinks to discover true isoforms. And their average F-score, you see, this is the native cufflinks, this is our NMFP plus cufflinks. So it's increased by about more than 10% in terms of F-score. So we think this is a very good result. We also coupled NMFP with a method I previously developed called SLIDE. And SLIDE, we tried two parameter settings. One will make SLIDE select fewer isoforms. One will make SLIDE select more isoforms. And then we use NMFP as a pre-selection step of SLIDE. And we see that in both parameter settings, we see, okay, especially for the green one, the more parameter setting, we see the increase is higher. For the fewer one, although it's not much higher, but still we see this increase. So this is on simulation data. And on real data, so we tried this on 74 real RNA-seq data in Fly. And here I'm just showing one real example. So here the top track shows the RNA-seq read, and this is the annotation. And this is what's found by cufflinks. So you see that cufflinks miss the last exon here in annotation, doesn't capture that. But if we use NMFP before cufflinks, we see that it actually contained those annotation isoforms. Of course, we found a little more, but at least it doesn't lose the last exon. And also for slide viewer, that's just the viewer parameter setting of slide. We see that, okay, this one captured this shorter one, which is not in isoform. But with NMFP, the shorter one is not discovered. So only this one is discovered. So we removed a possible false positive. And also for slide more, we see this. And the nice thing is that if you only use NMFP, without downstream method. So that means you are selecting the high confidence isoform candidates without further pruning. You can see that it's not actually that bad because it captures the superset of the annotation isoforms with a bit more. So for the additional ones, we will rely on downstream method to filter them out. And also, if you go back here, you can see this black line means NMFP alone. You see it's not that bad, especially the recall is very high. Yeah. Okay, so that's about the first method. And about the input and output of the method. The input is very simple. You just need the gene annotation file in GTF format, and you need multiple RNA-seq data files in BAM format. And the parameters, we have the default settings, but you have the freedom to tune them. And the output will be a GTF file containing isoform candidates. So as a summary, so the NMFP can effectively shrink the isoform search space to improve the performance of downstream isoform discovery method. And there are two remaining issues with NMFP. And so we are actually trying to speed up the algorithm. The first thing is about the parallelization, because we have to run NMF for multiple runs. And the second thing is about how to properly normalize the multiple data sets. So the source code and examples are available on my website. And so this is the paper about this method. And so for this work, I want to acknowledge Yu Ting, who used to be a summer intern undergrad student from China who worked with me. And right now he's a grad student in UC Berkeley. For the second part, which I'll go very quickly, is about another method developed by my group. And actually the leading author of this paper is sitting here. She's a student of mine. So first, for this project, we developed a new measure, transcriptome overlap measure, for finding transcriptome similarity of biological samples. We have the three aims. The first aim is we want to compare transcriptomes within or between different species. And the second thing is that we want to discover sparse relationships among many samples. So this sparsity is the key thing. We do not want to have too many correspondence, but we want to find something very simple, very sparse, if we want to discover some relationship among many samples. So for example, many tissues and cells in human or between human and mouse. And the third aim is to understand the conservation and divergence of biological processes in different species. So this is a consequence of using this method. And our approach has two parts. The first is that we want to identify associate genes that capture molecular characteristics of biological samples. So I'll define this term specifically in a later slide. And also, using these associate genes, we want to subsequently compare the biological samples by testing the overlap of their associate genes. So that's why we call this transcriptome overlap measure. So this is an illustration about the input data. So let's say you are interested in within species comparison of multiple samples in fly, Drosophila. So these are the samples and these are the genes. And this is the input gene expression matrix. Here the samples are 30 developmental stages. And using our method, first of all, in the first step, you can select associate genes for every sample. And this is just a bar graph summarizing the number of genes per stage. So here you can see that we are dividing the genes into different categories by the number of stages they are associated with. So the darker one means the gene is only associated with this one sample, and this means it's associated with 2 samples, 3 samples, and more than 3 samples. So you see that different from specific genes. Here, the associated genes can be associated with multiple samples if the samples are similar. And these are the basis for us to comparing the samples. And another thing you can see that is for the stages, here the developmental stages of fly that have higher transcriptional activities. So for example, the embryo, and these are the adult. So in these stages, you can see more genes are found as associated. So this is also meaningful because more genes are active. So using these genes, we can compare the stages. And this is the result. So you can see, using our method, this is the, so every entry here will print a score. Darker color means a higher score. You can see the diagonal pattern is very clear because here we're comparing stages ordered. So therefore, adjacent stages should be expected to have higher similarity. And this is the result if we just use Pearson correlation or Spearman-Rand correlation on the gene expression matrix. So Pearson correlation didn't do a good job for sure. But for Spearman, although you have this, darker colors in the diagonal, but you see it's not sparse. The relationship is not sparse. You need to apply a threshold to make them as 0, them as 1, to make it look sparse. But doing that is not actually enough. So this ROC actually shows that. We actually got this ROC from simulation. So the way we do the simulation is that we simulate samples with correlated associated genes. So that means adjacent stages have higher correlation in terms of their associated genes. And then on these genes, we use the real RNA-seq data here to get the gene expression value. So the only simulation part is the associated genes, but the values are from real data. And then so because we have that true correlation matrix, we can binarize that to make it just 0, 1 label. 1 means that 2 stages should be mapped, and 0 means they are not mapped. So mapped means high similarity. And then we use these matrices as features to classify the stage pairs into 0 and 1. And this is the ROC we get. So you see, our method is the top performer, and then Spearman is the blue one, yellow is the Pearson one. So you see that here we have much better AUC, almost 1. The reason is because what? It's because our method is a transformation of the gene expression matrix and make it as a better feature for classifying stage pairs into 1 or 0. And that's why we perform much better. And that's why I'm saying the advantage of our method is the sparsity result. Okay, and for between species comparison, here you have 2 species. And in this example, we have fly here, we have worm here. They have different number of stages, different genes. And you also need, for our method, you also need the ortholog relationship. So this is the fly gene, this is the worm gene, and every row represents the ortholog, a fly-worm ortholog. So this is the result of our method. You have the worm stages, fly stages, and this is the result. So here the comparison is based on the number of shared orthologs in their associated genes. So between a fly stage and a worm stage, we take their associated genes and count how many of them are orthologs. And then we test whether this is significant. So this is from Pearson correlation, Spearman correlation. So you see the signal of these 2 is not as clear. And here we see a very nice pattern because as worm stages proceed, we also see fly stages proceed in this pattern, and we see another pattern. And another important thing is here, you see this breakpoint in the fly stages actually correspond to metamorphosis. So it's something very interesting because during metamorphosis, all the fly cells will be dead, and new cells will be regenerated from stem cells. And actually, we found that. Okay, so this is the workflow of the package. In this package, essentially, you need to input gene expression matrix. And you have the option to let us select associated genes for you, or you can tell us which genes you think are important. And then with those associated genes, we will find the overlap. So we will do this, the within species TROM, between species TROM to give you the heat map about the mapping. And also we will do the GO term analysis and output the GO terms to help interpret, why these 2 stages are mapped to each other, which genes lead to this mapping, and what their functions. Okay, so as a summary, so this method is a useful measure for discovering sparse relationships among many samples. And we have an R package for this available on CRAN. And so this work was actually motivated by a previous work I had published on genome research, but that was an analysis paper. Then we formalized this method for a lot of technical details and also a lot of statistical properties in the paper, which is currently on archive and will be in press soon. And I want to acknowledge my students. The first student, Wei, she's sitting here, and she's currently a PhD student in our department. And also an undergrad student who is currently in UCLA, but she will join our department in this fall. Okay, thank you.